Okay. So I would like to introduce our presenter. Cathy Cripps is a professor in the Department of Plant Sciences and Plant Pathology at Montana State University in Bozeman. And she teaches courses there on the biology and ecology of fungi. Cathy's research concerns the systematics, diversity, and ecology of higher fungi in mountain systems, in forests, Arctic alpine habitats, and also on disturbed sites, particularly in smelter impacted soils. Cathy specializes in the mycorrhizae of aspen, whitebark pine, alpine plants, and those that can be used in reclamation and reforestation. She's the author of numerous technical journal articles, but she is certainly not a snooty academic. If you watch public television, chances are good you have seen her on Montana Ag Live. And she is the author of this book. You can see that. Essential Guide to Rocky Mountain Mushrooms by Habitat, and that is for us lay people. And Cathy has a passion for collecting wild mushrooms, and she's currently categorizing the species that are found in Montana. So because there are so many people on our Zoom tonight, if we have questions in the middle of the presentation, there's a real risk of sort of getting diverted away from the presentation and getting lost while it's going on. So I'm going to ask that you type your questions into the chat box, probably addressed to me, but you could also address them to everyone. And I will carry them on to Cathy at the end of the presentation. And if there are several similar questions, I might paraphrase them into one. So please don't take offense if I don't read your question verbatim. So I'm gonna mute, stop my microphone. Cathy, I think you should be able to share your screen now.
All right, let's see what happens. Okay. Did that come up?
It certainly did. Thank you.
Whoa, all right. That's great. Do you want me to just go ahead and go? Are we all ready here?
Go ahead and go.
Okay. All right, I'm just trying to see what I need to minimize here. Okay. So, good evening, everybody. I'm glad you could join us. I see a lot of names that I recognize on there. I want to give a shout out to all those folks that I know. I want to thank the Native Plant Society for inviting me, and especially Gretchen Rupp. We ran into each other on one of the trails around Bozeman, and she asked me if I'd give a talk. So here I am. I'm talking about one of my favorite subjects, the mycorrhizal web in Montana forests. So what I want to do this evening is, first of all, give you a little bit about the basics of what are mycorrhizae, sort of Mycorrhizae 101, and then kind of wade into it slowly, looking at some ecology of mycorrhizae in Aspen Forest and then in Conifer Forest. And these will be things that you can observe as you're walking through the forest. And I'm hoping this sets the stage, get you used to the idea of mycorrhizal fungi and some of their patterns out in nature, so that then we can move to some of the topics that are kind of hot topics today, primarily from Suzanne Samard's research, such questions as, do trees communicate? Do trees help each other? Do mother trees nourish their young? Can trees warn each other of insect pests? So we'll get to those questions after we do the warm up here. So let's go ahead and start. And here we have a Montana forest. and I'm sure all of you have walked through one. And the trees are obvious. Someone is not muted. And the understory is easily seen when there is one, the animals, the insects. But for mycorrhizal fungi, it's a little bit more serendipitous when you run across a mycorrhizal mushroom, such as this red one shown here. And that mycorrhizal mushroom is basically the tip of the iceberg, the tip of everything else that's going on below ground that we can't see. So it's above ground evidence that mycorrhizal fungi are functioning. You know, forests are 80% of the biomass on Earth, and on average 25% of that is below ground. So we're missing a lot of the ecology of our forests when we leave that out. So that's what we're going to talk about today. Well, here's what E.O. Wilson tells us. Most life on land depends ultimately on one relationship, the mycorrhiza, the intimate and mutually dependent coexistence of fungi and the root systems of plants. So think about that. Most life on land depends on mycorrhiza. Now, why does he say that? Well, plants are the base of the food chain, and plants depend on mycorrhizal fungi. Now, I've photoshopped in here the mycorrhizae on this root system just to show you that they're down there and that they're hanging off the root. And when they reproduce, then they send up a mushroom. I'm going to see if I can hide that thumbnail. There we go. That helps me. Okay. So that is, again, the above ground part of the mycorrhizal system. Now, let's start very simply, just with a pine seedling. And the shoot comes up, the seed germinates, and the root system goes down. Now, if that was in a container or a pot or the greenhouse or the garden, what would you have to do to keep that going? Fertilize it, right? You'd have to add fertilizer to keep that seedling going. Well, obviously, that doesn't happen in nature. So let's see what does happen in nature. And this is going to be a little bit disruptive because those links don't quite work on this. So I'm going to try to share something else. I'm going to try to stop the share, and then I'm going to try to share another screen. And, oh, it's not there. I don't think this is going to work. That's really too bad. Hang on just one second. No, we lost it. Well, I'll just share this one and just, I don't think that's going to work. No. Hang on. And I hope I don't get thrown off. If I get thrown off of this, then let me back on again. Okay, now let's see if I can try that again. And new share. Let's try this. Okay, can you see that?
That worked, Kathy. We can see.
Did that one work? Yay. Okay, so let me start this little video. And here we see, this is a time lapse over many, many, many weeks. And what you're looking at are pine seedlings germinating. And here the seedlings are coming up. They still have the seed coat on. The radicals are going down. They circle around. That's called nutation. Now the seed coats are coming off. The needles are popping out. And that root system is going down. There's a little bit of white fungus here and a little bit of white fungus here. And now we're going to look at a little bit of a close-up of what's going on below ground. And what you're seeing in time lapse are the actual roots of the young pine seedling growing. And notice all the white threads in the soil. Those threads are the fungus, and they're covering the roots and making them white. So all of the white threads cover the new young roots of those seedlings. And sometimes they produce chemicals that actually make those roots branch in a unique way. Here it's branching it in a V shape. And in the end, here's what you end up with. This is a mycorrhiza. And if you look closely, it's a set of roots, branched roots, covered with that mycelium of the fungus, totally covered. Now I'm going to stop this, and I'm going to hope I can get back to go. And I'm going to stop that share. And hopefully I can pop back onto here. And okay. Okay, are we back to go? The screen on?
We are.
All right. Okay. So what you've got going on here, what you just saw in that little video, is the fungus, mycorrhizal fungus, in the soil had joined up with the roots of the plant. So that oval down here is what an actual mycorrhiza is all about. It's part plant, the roots are underneath. It's part fungus, that white layer over the top is the fungus. Now, if you look up at the seedling on the top of that, those little tiny balls right on the root are those mycorrhizae. So what's going on here? And then the rest of this is mycelium going out into the soil. So the plant, through photosynthesis, makes sugars, and it sends the sugars to the roots. And that's what the fungus is living on. The roots are kind of leaky, and they can take as much as 20, 30% of the carbon from the plant. And for a long time, as you might imagine, we thought that these were parasites hanging on roots. But it turns out that's not the whole story. All of these threads, the mycelium, that's the body of the fungus. This is what an actual fungus looks like. All of those threads go out into the soil, scavenge nitrogen, phosphorus, and other nutrients, and shoot it back through that mycelial system back to the plant. So it's a mutualism. The fungus gets the sugar, and the plant gets fertilized. Mycorrhizal fungi, bottom line, Mother Nature's fertilizer. And when the roots or the mycelium grows up towards the top, they might produce a mushroom, and you see evidence. So that's the other end of the mycorrhizal fungus. So there are a lot of types of mycorrhizae, and I'm just going to talk about one. The type that you just saw forming in that little video clip is called ectomycorrhizae. Ecto because the mycelium's on the outside, and you can dig up into the soil, in the duff, and look at fine roots, and you can actually see these on roots because they're on the outside. And these are the fungi. This is the type of, I should say, mycorrhiza that forms with a lot of our forest trees. So aspen, alder, willow, cottonwood, birch, and our conifers as well. So I'm just mentioning, we're not going to talk about these today, but there are other types. The other main one is arbuscular. It's with grasses, forb, shrubs. So there's a whole nother story here, and some other trees. And then some other types listed down on the right. If you can't see them on your screen, ericoid, orchid, monotropoid, etc. But I'm just going to talk about the ones with trees. And I may not say ectomycorrhiza, I may just say mycorrhiza. And one other thing, I just wanted to make sure everybody was aware of, that just because you see a mushroom under a tree does not mean it's a mycorrhizal mushroom. Different species of fungi have different lifestyles. you basically need to know the genus of the mushroom to know if it's a mycorrhizal mushroom or not. So this Amanita is mycorrhizal with cottonwoods. Because it's an Amanita, I know it's mycorrhizal. Other fungi are decomposers, such as this delicious oyster mushroom. Notice it's up on the side of a cottonwood. It's not even connected to roots. But there are other mushrooms that are on the ground that are decomposers, so that are not mycorrhizal. And there are a few mushrooms that are actually pathogens, such as this honey mushroom, armillaria. Now it's connected to roots, but it's producing a root rot, so it is not a mycorrhizal fungi. So we're just going to talk about the mycorrhizal fungi here. Okay, so those are the basics. That's kind of the warm-up. Now we're going to take a walk first through an aspen forest and then the conifer forest. And the reason I started with an aspen forest is because this is how I started my research. Long ago, I really became interested in aspen forests and learning what mycorrhizal fungi were in aspen forest. Well, why aspen forest? Because it's a simple system. It's one tree species. And not only that, these are all what look like separate trees are actually connected below ground because aspen forests are all one big giant root system. So this is a clone. This is all one big organism. We know possibly Pando in Utah is the world's largest organism. So with aspen, is there just one type of mycorrhiza below ground? Are there a few? Are there a lot? When we talk about mycorrhizal webs, I've heard people talk about it as if it's all one big giant fungus down there that's all connected. Well, that's what I wanted to find out, what's down there. So I picked some sites, some of you might know these, and I picked 3 sites on purpose because they have different soil types. So besides wanting to know what fungi were down there, I wanted to know if soil made a difference. So I have the East Ridge of Butte, really, of course, acidic soil, sandy, from the smelters that were functioning in the past. Cinnabar Basin down in Gardner, kind of a gravelly loam soil. And then some of those upland forests in the Tetons have a really calcareous, silty soil. So would I find all the same fungi on all these sites? What kind of diversity? So I was just getting started. So how do you study mycorrhizal fungi if they're below ground, right? Well, here I am digging a soil pit, and this was primarily just so I could analyze the soil. But I also got to dig around down there and see what was going on, all kinds of interesting things. So the mycorrhizae are mostly in that top organic layer. They're a little bit deeper in the soil on the Butte site, but in general, mycorrhizae are up in that organic layer and a little bit down into the mineral soil. So what I did was collected the fruiting bodies, the mushrooms, of mycorrhizal fungi for several years so I could see what was functioning, and I knew I could find these above ground. Then I did soil cores to look what was on that, see what was on the aspen roots. And some of these, you can actually recognize them on the roots just by the morphology. This was before the era of DNA. Now they can match it up with DNA. So make a long story short, what's down there? What did I find? Well, lots of hidden diversity. Over 40 different species of mycorrhizal fungi. And I think since then, in other aspen forests, I've really increased this number. So lots of diversity of fungi down there. And it turned out that most of them were only on one soil type. Some of them were on all 3 soil types, but some were just on one soil type. So soil really did make a difference. And here you can just see the diversity of mycorrhizal fungi that are in aspen. In that bottom row, you can see the diversity of some of the mycorrhizae that I dug up. And I just want to make a point here. This is a diagram that mycorrhizal researchers often show. And what this means is the target is the formation of mycorrhizae. So you need the right plant, in this case, aspen. You need the right fungus, one of those that I just showed you. But there's another filter. You need the right soil type that these fungi can handle. If all of these, and plus a few other things, climate, etc., are working, then mycorrhizae form. So it's not as simple as fungus tree. You have to have special conditions. And I found out something else, and this, I hadn't realized this. This kind of surprised me, that tree age made a difference. Some of those stands at Butte were younger, those aspen stands, because the smelter knocked back the vegetation and the aspen were slow coming in. So they were younger stands. And what I found there was a lot of what I'll call weedy mycorrhizal fungi, weedy species. They didn't need organic matter. They didn't need very good conditions. They just come in quick on young seedlings, and they're often not very host specific. It's like weeds coming in a field after a field is disturbed. What comes in first? Weeds. They don't need much, right? Well, these are weedy mycorrhizal fungi that don't need much. Early colonizers. But as the forest develops, you get other aspen leaves fall, they decompose, you get more organic matter, shade, that holds the moisture, good conditions is developed. And so a diversity of mycorrhizal fungi can come in, and brought in by spores, brought in by animals, which I'll talk about later. And then here's an important point. So all of these fungi below ground with their mycelium, they're not holding hands with the mycelium. They're competing with each other. They're competing for space, for nutrients on the tree roots. And the good competitors eventually, as the trees get older, kind of win out. And they tend to be more host specific because they have a tighter connection to the tree. So these are the late colonizing fungi. So there's a whole succession. There's patterns in space and there's patterns in time for the mycorrhizal fungi, is kind of the point that I'm trying to make here. So why so many different kinds of fungi? Well, not all mycorrhizal fungi do the same thing. They do have that basic functioning of getting carbon from the plant and feeding it nutrients and fertilizing it. But different fungi have different specialties. Cenococcum helps with drought tolerance. Paxillus prevents heavy metal uptake in aspen. We saw a lot of this on the Butte site. There's a lot of copper in that, as you might imagine, in the Butte soils. And those heavy metals, the copper is stopped at the mycorrhizae so it doesn't get up into the aspen, so that the aspen can actually grow on those heavy metal sites. That's why some fungi are used in reclamation. And there are others, such as the orange cap boletes here, the lexinums, provide a lot of nitrogen to the aspen. So every fungus has its own ecology. I'm just mentioning a few. So something to keep in mind. Oh, and I just put this in for fun, a little break here. Ecological connections of really different organisms. And I bet most of you in the audience could connect up the first three. I'm adding one more. So wolves were introduced to Yellowstone. And when they were introduced, the elk population declined, as expected, because wolves are predators. But what was less expected is that aspen and willows proliferated because the elk were not browsing them as much. Well, in my world, in my mycorrhizal world, it was not just aspen and willows proliferating, but whoa, it's a whole new set of mycorrhizal fungi that are coming in. And these mycorrhizal fungi function a little bit differently than those in the conifers. It's a different set. So who would have thought that introducing wolves to Yellowstone would make a difference in the microbial functioning of the soil? So some connections here. Okay, let's switch over to conifer forest. This is my husband, Don, walking in the conifer forest, somewhere up highlight. So what are we going to see in conifer forests for mycorrhizal fungi? Well, first of all, there's a lot of edible mycorrhizal mushrooms in conifer forests. I'm sure some of you have collected the golden chanterelles. Why do we go to conifer woods to find the chanterelles? Well, because they're mycorrhizal with the conifers. Simple as that. And the same with the king bolete, is a mycorrhizal mushroom, eminently edible, king boletus edulis, and hawkwings, Sarcodon imbricatum, another edible mycorrhizal fungus. So you do not see these in aspen forest. Now, what I'm not showing you is that whole plate of all the different mycorrhizal fungi in conifer forest, like I showed you with aspen, because there are hundreds of different mycorrhizal fungi in this forest, and they're mostly different from the ones in aspen forest. There is some overlap, but a lot of them are different. So once again, we see patterns with different kinds of forests, as well as those seasonal patterns and age of tree patterns. Those kinds of patterns are what I'm trying to get across. And this is worth taking a closer look at. The early colonizers of conifer forests are different from those in aspen forest. Some of them, so I shouldn't say that, some of those are the same, such as some of those weedy species I showed you. But these you will not find with aspen. These are only early colonizers with conifers, and it's worthwhile learning these names, slippery jacks and pogies, because they're going to pop up, so to speak, throughout the rest of the talk. So slippery jacks are small mushrooms that have a sponge layer underneath, and pogies, or rhizopogon, grow below ground. These are dug up. They look like little potatoes or tubers. And when you cut them open, the spores are inside. These are related to each other. DNA tells us they're really closely related to each other, even though they don't look anything alike. The pogies have developed an underground habit, and the slippery jacks are above ground getting their spores out through the wind. If you dig below ground, I hope this shows up on your screen, the mycorrhizae of pseudoloids are somewhat distinctive. They're highly branched. Some of them are called coralloid or just multiple branched. You can pick them out if you dig around the slippery jack or beneath the pogie, or you see pogies popping up from the soil. You can dig down and see these highly branched mycorrhizae below ground. That becomes important later on as well. So the slippery jacks get their spores out by the wind. But what about pogies? They're underground and the spores are inside. So how do those spores get out? Well, some of these are called false truffles, not the true truffles, but false truffles. And they've developed unique odors. And those unique odors attract mammals, which eat them and spread the spores. How does that work? Here's a nice little squirrel that posed for me on a whitebark pine while he was eating a fungus one day. I have lots of photographs of this little guy, all the way till to the end of the fungus too. So squirrels eat pogies, rhizopogon, and the spores go through their digestive tract, come out the other end, and the animals are moving around and spreading those spores. And there's research showing that those spores are still viable when they come out the other end. This is how the rhizopogon gets around. I think one thing we might need to remember in whitebark pine forest, sometimes squirrels get a bad rap because they go up and get the cones from the whitebark pine and sometimes beat the birds to it, right? Which are spreading the seeds. Well, the squirrels are spreading the mycorrhizal fungi, which help the trees grow. So you have to keep that in mind too. And the large mammals will also eat pogies and spread the spores. I've watched deer eat pogies. I've caught them in the act and checked to see what they were. And grizzly bears eat them as well. I had a call once from the grizzly bear team in Bozeman, and they asked me, what are the grizzly bears eating that they dig down with their noses and pull them out and eat them? And I said, well, bring me one and I'll tell you what it is. And they said, well, the bear's nose is so good that it's getting everyone and we can't get one. And I said, well, bring me some grizzly bear scat. And they did. So they brought me 30 samples of grizzly bear scat. And I thought, oh boy, now I'm looking for a needle in a haystack. Spores are 10 microns long, and finding a spore in grizzly scat, I thought, well, this could be tough. And I thought, well, I might have to filter it and go through all kinds of scientific things. But I thought, well, I'll just take a pinch of it, put it under the microscope and see what's there. Lo and behold, all of these little ricey structures here are the spores of rhizopogon. Just thousands and thousands of spores throughout the scat. And every sample I took had spores in it. We could pick out some of the groups. They were all rhizopogon spores. So those bears were really good at zeroing in on that particular odor and digging these up. So it's also spreading. The grizzlies are also spreading these spores. Now think about this, that the bears are spreading the mycorrhizal fungi, which are eventually going to connect up with seedlings of whitebark pine and other pines, which if they're whitebark pines, then they're going to be feeding on the nuts. So here's one more connection. They're actually bringing the mycorrhizal fungi to the seedlings. Okay, let's go back to the slipperyjacks, the Swillis. These are a little bit more host specific than the Pogies. Pogies can be on several different kinds of conifers, but not so the rhizopogon. They're host specific. So if you're in a larch forest and you see a mahogany colored slipperyjack, probably Swillis grevillei. Douglas fir, that pink scaly cap, Swillis lakei. Whitebark pine has its own Swillis, Swillis americanus or Swillis sibiricus. Lodgepole pine has its own Swillis. So if you think about this, these are the kinds of mycorrhizal fungi that are not going to be able to connect up different tree species. They stick to their own host. So we're not going to have a giant mycorrhizal web of all of these connected below ground. Fungi don't connect their mycelium below ground. They compete with each other below ground. Something to keep in mind here. Oh, just for fun, another break. So squirrels, squirrels dry mycorrhizal fungi in the trees for winter. These are just some of the photos that I've snapped of them. So far, I have not found a non-mycorrhizal fungus up in the trees brought there by squirrels. So they're pretty good taxonomists. A lot of them really like russulas, which is a mycorrhizal fungus, and they'll cover a whole tree with russula and dry them for the winter. It must be the odor that they're cluing in on, but this is another way the spores can get around if they're hung in trees. And someone sent me this photograph of elk eating big white mushrooms late in the fall in Yellowstone National Park and wanted to know, well, what are the mushrooms that they're eating? And so they sent me a photo, and it turns out that this russula species, Russula brevipies, quite common around here in the late fall, is a mycorrhizal fungus, but we don't know much about this system. If anyone sees this phenomena, document it for me so we can see if this is typical or atypical. Okay, so as you might imagine, mycorrhizal fungi have been used in restoration, and I was involved with mycorrhizal fungi in the restoration of whitebark pine, which is of course our high elevation 5-needle pine that's in peril. The populations are being decimated by white pine blister rust and mountain pine beetles. This is a photo from Glacier Park of a ghost forest. The boles still stand, but the trees are dead. I often wonder how long that mycorrhizal system stays in place below ground after these forests die. But there are huge restoration efforts underway. Forest Service, BLM, Parks Canada is planting hundreds, thousands of seedlings to try to replace these forests. So how I got involved with that is from the mycorrhizal standpoint. A lot of the seedlings were dying when they were outplanted. So would adding any kind of mycorrhizal fungus help? We found that Suillus sibiricus was our fungus. It formed mycorrhizae very easily in the greenhouse. We found that the seedlings were 60% larger. They had a lot more nitrogen in them when they were outplanted if they became mycorrhizal. If anyone's interested in that technique, you can just Google, there's a little thing that says Cripps inoculation of whitebark pine or something like that. And a video will come up showing you how we do that. And this is just one of our latest sites is over in the Gravelies where there were a lot of forest fires. Whitebark pine are often planted on burns. And we're monitoring, do the mycorrhizal fungi help, aspect, elevation, a lot of other site factors to see what helps for the survival of these seedlings. We found out now that it's a 2-part process. Inoculation is one part, actual colonization is the second part. So we're working out the bugs for monitoring in that. Okay, now here's a slide that I need to tell you about before we get to the third part of the talk, the promiscuous mycorrhizal fungi. And the promiscuous mycorrhizal fungi are found on all different kinds of trees. And there are quite a few of these. Cenococcum, I've mentioned before, it's found on aspen, all of the different conifers, birch, lots of different tree species. Piloderma, found on lots of different species of conifers. The lephra, found on aspen and lots of different conifers, and then there's some others below. The reason I mention these is because the promiscuous fungi have the potential to connect up different species of trees with each other. So they don't have to stick to one kind of pine or just aspen. They can start to make connections between trees below ground. Okay, so that sets the stage for this last part of the talk. Now you know something about mycorrhizal fungi. And this is Suzanne Samard's research primarily that I'm going to be talking about now. And I imagine or hope that quite a few of you have read her book, Finding the Mother Tree by Suzanne Samard. She's a colleague. I don't know her really well, but she is a colleague. And this is becoming a bestseller. So if you're interested in mycorrhizal fungi, you might be interested in reading the book. It has some of her life in there as well. So it's a good read. I'll put it that way. It's a good read. But Suzanne started asking, she's a forester in British Columbia. She's done a lot of extraordinary research. And she started thinking about forests. She started thinking about, are trees actually communicating with each other? Is there a wood wide web? Do trees help each other? Do mother trees nurture their young? And more questions that she was asking in the kind of way that made it really exciting and sort of shifting our paradigm of how we thought about forests. So of course, you know, mycorrhizal fungi are going to come into play here. So here's what Suzanne's research is all about. Here's what she's doing here is pulling down a branch of a large conifer tree shown in the back and covering it with a plastic bag. And then they're shooting in carbon dioxide. And of course, trees need carbon dioxide for photosynthesis, but this carbon dioxide is a little bit different. It's labeled, it's a slightly radioactive so that you can follow where it goes. Actually she uses a Geiger counter. So the carbon dioxide goes into the needles. It's used for photosynthesis. You can track that carbon as it goes up the branch and it goes down the big tree trunk. It goes into the roots of the tree where maybe it's going to store some of that carbon. But lo and behold, this other photo over here, this is photoshopped. They put in these kind of fancy lit up places. But what they're trying to show you is that the carbon has come down from the main tree into the roots of the main tree, past that into the roots of the young trees around there, and even up into some of the young trees. So it's getting around. There are connections here. And this was quite a surprise. And I think she did several experiments, and this is absolutely what goes on. That diagram shows you tree roots of two trees connected by a ectomycorrhizal fungus, and the carbon going from one tree to the other, and maybe it's going from this one to the other way. In this case, you have two different tree species. So you're thinking of those promiscuous mycorrhizal fungi to make that connection. So here it looks like a complicated diagram. It's not that complicated. It's actually, Suzanne's question is, there a wood wide web? And how could she get to that? So she showed the diagram with the Geiger counter, but here they really delve into it. So what you're looking at with all these dots is basically a Douglas fir forest. And each one of the dots represents a tree. The big dark dots are the older trees. The smaller light colored dots are the younger trees. It's just as simple as that. Except they were using kind of a fancy DNA analysis. It's called microsite analysis. What they could do is tell the individual trees by their DNA, not just the species of tree, but they knew they were all Doug fir, but they should have numbered these. This is like tree number 1, tree number 2, tree number 3. They could tell by the DNA, it's tree number 1. So what they did was then dug into the soil and dug up mycorrhizal roots, and they were looking for those special kinds of mycorrhizae that Pogies make. Remember I mentioned that highly branched? Well, they dug those up, and then they looked at the mycorrhizae DNA. Did that root match tree number 1 or tree number 2? They could match up all those roots to the appropriate tree. Well, fungi also have individuals. This could be all one fungus underground, but typically it's many, many, many different individuals, many different mycelia down there, actually all competing. Well, they could figure out what fungus, what individual fungus was on a tree. And that's what all those black lines are. They could tell how those trees were connected for 2 different kinds of pogies. They only did the pogies. So yes, there is definitely a wood wide web. All of these trees were connected. Rather astounding. And the different individuals of the fungi connecting these different trees. Mother tree down here, just because it has all these black lines connected to everybody. And then something else to think about is that they only did the rhizopogons. They only did 2 species. There are a lot of other fungi down there, mycorrhizal fungi. So then the web gets layer on layer on layer. So it gets rather complicated. So yep, there's a wood wide web. And now we know what flows through that web. Carbon, nitrogen, phosphorus, water, chemical signals move through this mycorrhizal web from one tree to the other. So yeah, the trees are communicating with each other below ground through that mycorrhizal network. Well, how are nutrients actually flowing in that mycorrhizal web? Well, just to make it simple, I'll put it this way. They flow from source to sink, and basically, if there's more of something here on the left, say, it flows to where there's less of something. It's like a gradient, like a downhill gradient, if you want to think of it that way. Simple as that. So if you have more carbon on that tree on the left, it flows through the mycorrhizal fungus to the tree on the right. And same for nitrogen, phosphorus, water, stress, defense chemicals. That's what makes them flow. So if we put that all together in a forest, now what? It's complicated, right? So here we have an actual forest, not just a diagram. You have big trees and small trees and different species of trees. So what's going on here? Now we know there's a mycorrhizal web down there. Well, there's still competition going on between these seedlings for light and water, soil, space, nutrients for mycorrhizal fungi. There's still competition, but Suzanne has tried to shift our paradigm some from solely competition to also cooperation, because these trees are connected, and in a sense they are cooperating with each other as a whole. Some of these, if it's between different species, you probably can't tell, but this is a whitebark pine seedling in front and lodgepole pine seedlings behind. It would take some promiscuous fungi or some fungi that are can connect up with any pine to connect those different species. The lodgepoles can have their own mycorrhizae. If this whitebark pine has a Sewilla sibiricus, it cannot share that with the lodgepole pine. So it has an advantage with that fungus. But promiscuous fungi on any tree, right? Well, like promiscuous relationships, there's also called dangerous liaisons. So if a tree taps into that mycorrhizal network, which way are these things going to flow? This carbon, this nitrogen, is the tree going to lose or win? And some do lose, some do wins. And so basically you have to think about which way are things flowing and who benefits, but it is all connected in some way in different ways. Yes, the wood wide web below there. what about this question? Do mother trees nurture their young? This is one of Suzanne's questions. That's from her book, Finding Mother Tree. So we have a mother whitebark pine tree in the back, and we have a seedling whitebark pine in the foreground. So if you're envisioning the root system of the mother tree hung with mycorrhizal fungi, and those mycorrhizal fungi growing towards that seedling to nourish it, that is the wrong concept. That's not what's going on. This is what's going on. So now think about the mother tree with the root system going out and all of the mycorrhizae hung off of that root system. If the seedling lands within that mycorrhizal web, it can tap into that mycorrhizal web. And yes, carbon will flow from the mother tree to the baby tree. Yes, the mother tree is nourishing that baby tree because it tapped into that mycorrhizal web. So it's a little more serendipitous. Of course, if the seed of another tree species landed in that mycorrhizal zone, if the right fungi were there, it could tap into that as well. And who knows, that could be a father tree. So can other plants take advantage of that mycorrhizal web? What about, we used to call these parasitic plants, the word now is mycotrophic. So striped coral root, pine drops, can't photosynthesize, they don't have green leaves. So how do they make a living? They tap into that mycorrhizal network and they get their carbon for growing from that mycorrhizal network. If you carefully dig below these plants, each one has a ball of mycorrhizae down. I'm not recommending digging all of these up by any means, but if you want to take a peek, you might be able to just dig down a ways and take a peek and see all of these mycorrhizae are for the purpose of bringing that carbon into the plants. Now, it could also be that later on in the year, these plants die and then they contribute carbon to the mycorrhizal web, so it can flow the other way. Some of the latest research, interestingly enough, has showed that what they're tapping into is rhizopogon, Pogies, and Pseuilus mycorrhizal systems between the trees and those fungi. So there's some fascinating, people are doing DNA work and finding out that's the system that they're tapping into. Okay, so can trees warn each other of pests? Well, a lot of this research was done in the greenhouse so they could have controlled conditions. So I'm kind of extrapolating to the field here, but they did, this was Suzanne Samard and her crew again, looking at very controlled experiments showing that if you had spruce budworms defoliating trees, I'm not sure how old their trees were, that yes, defense chemicals, stress signals from the defoliated tree can move through the mycorrhizal network from the stressed trees to trees that have not yet been stressed, and they can prime their immune system. The activated defense genes get them ready for those pests should they come. Now, trees also send out volatile chemicals through the air, and they do the same thing in a sense, but in the lab, they could control for those volatile chemicals in the air, making sure that that's not what was causing the reaction in the unharmed trees. So it was absolutely moving through the mycorrhizal network. And then finally, this is Suzanne's Mother Tree Project. And you can find this on her website. This is where I took these photos from. And this is about clear cutting, leaving legacy trees, and the mycorrhizal web. And here's, of course, typical clear cutting. She's found that leaving single trees, or better yet, in some cases, groups of trees, helps preserve that mycorrhizal network. Now that you know about it, you understand where she's coming from. And she has shown that it does improve natural seedling regeneration. And her papers down below, she actually has actual numbers for her system. I think she was working with Douglas fir, and she has the actual numbers of trees per hectare that she would leave to maintain that mycorrhizal web. So some of the forestry practices are beginning to be shifted up in British Columbia. Of course, there's also resistance to that as well, but she's has some pretty solid research that she's shown. Okay, so as we wind down here, so now you know something about the mycorrhizal web, and I think the next time you go into a forest, you can think about everything that's going on below ground and that the trees are communicating between each other. They're connected. And as E.O. Wilson says in another way, most terrestrial ecosystems are predicated on the existence of microscopic fungi on roots. So this is something to change your view of the forest the next time you walk through there. And then finally, here's some resources for you guys if you're interested. My book, Gretchen showed this. I thank you, Gretchen. Rocky Mountain Mushrooms by Habitat. It's not a typical mushroom identification book. It takes 12 different habitats. If you know what habitat you're in, you'll learn something about the fungi in that habitat, and you'll be able to recognize a few of those, from alpine to burns, the different kinds of conifers, aspen, cottonwood. It's more for the casual mushroomers. Of course, Suzanne's Tree, Finding the Mother Tree, as I mentioned, if you haven't read it, it's a good read. Here's another one. This is The Entangled Life, and it has a pretty good section on mycorrhizal fungi. Mycotrophic wildflowers might be a good one for this group. The Forest Service has a website. I think if it's just mycotrophic wildflowers, I think it'll come up, Forest Service. And then I mentioned my inoculation of whitebark pine video. I just finished a Mushroom Hour podcast. You can just go to Mushroom Hour podcast and my podcast will come up. So now you know about mycorrhizal fungi. That's the tip of the iceberg. And I just want to say thanks to the Montana Native Plant Society for inviting me. So I am going to, I think, stop sharing if that's what Gretchen would like me to do. And we'll just see about questions. And am I back?
You're back.
Okay.
Thanks a million, Cathy. We do have quite a number of questions. You want to get a drink and I'll start bombarding you with questions.
Well, I have water right here.
Right.
I'll wait till wine for later.
Oh, no, I wasn't suggesting you start tippling. So I'm going to go through the questions sort of in the order that they came, which is kind of the order of your presentation, of course.
Yeah, that would be good. Yeah, let's try that.
So Myrna Peters has asked, is it safe to say that if the mushroom is coming out of the trunk higher than the ground, then is a decomposing fungus? But if it's coming out at the trunk or the root flare, it could be either a decomposer or a mycorrhizal fungus.
In general, yes, I would say 90% of the time, maybe 95% of the time. But I have seen mycorrhizal fungi that get in under the bark for some reason if there's a crack, and you can find them higher up the tree. But as a general rule, she's absolutely correct. Yeah.
I have actually seen oysters coming out 20 feet up.
Yeah.
Michelle Barber has asked, can we sprinkle mycorrhizal spores onto soil to help transplanted aspens and pines? And if so, where or how?
Yeah, there's not a lot of research showing that just sprinkling the spores would really help. I mean, I suppose it could, but there's just not a lot of research to back it up. What there is research for is actually extracting the spores from the mushroom, making a spore slurry, injecting it into seedlings, and then planting them. It takes 2 to 3 months in the greenhouse for mycorrhizae to form, and then you outplant them. And there's not really a lot of research that shows sprinkling the spores helps. Not to say that it doesn't, we just have no research showing that.
Interesting. Robert Fimbrell has asked, do mycorrhizal fungi form symbiotic relationships with nitrogen-fixing organisms such as Frankia, where the Frankia provides nitrogen to the fungus, while the fungus provides nutrients and water or some level of protection from pathogens in the soil?
I'm not exactly sure how all the connections work, but alder have nitrogen-fixing bacteria and ectomycorrhizal fungi both. So there's all three in that system, but I'll have to tell you, I don't know the exact relationships between all three of those. I'm not sure how that works, but alder is ectomycorrhizal and it has nitrogen-fixing bacteria. So that's one case where you can have 3 organisms, which is pretty fascinating.
3 different kingdoms, that's cool.
Yeah.
So Beth Madden asked a question about the radioactive tracer experiments. She asked whether the radioactive carbon actually becomes sugars that are transmitted into the younger trees.
Well, sugars at first. There's also some research talking about movement of carbon as amino acids. And I have to say that I don't fully understand that. So sugars in the first part, but then it may not be, there may be something that I don't know for that transfer below ground.
Andrea Pipp has a couple of questions. They have to do with sort of handling mushrooms. She asked, how easy is it, or what's the technique for matching a fruiting body to the mycorrhizal mycelium? Because the mycelium seems pretty fragile. And then connected to that, how do you preserve the mycelium? You have the fruiting bodies in the herbarium. Is a mycelium attached?
It's usually not the mycelium that you're matching up, it's the actual mycorrhizae. So you have the mushroom above ground, and then you have all the threads, which are the mycorrhizae. We don't preserve those, and we don't try to match. All the mycelium looks the same. But when the mycelium covers the roots, that root tip has a certain kind of morphology. It might be white or yellow, it might be fuzzy, it might be smooth. And sometimes we can match the morphology of the mycorrhiza with the mushroom. Those orange cap mushrooms that are mycorrhizal, when you cut them in half, they stain blue. When you look at their mycorrhizae, if you bruise them, they'll stain blue. And so there's matching, you could do that. And now people are using DNA. I do it the old-fashioned way. I use the morphology, but now people are doing the DNA and matching it up. So they can do a more efficient job than I can one by one. Does that make sense? I hope.
So Katie McCann was interested in the link to Suzanne's paper that you showed quite late in the presentation. You know, I don't have the URL. I can only suggest that you bring up this presentation on our YouTube, the Valley of Flowers MNPS YouTube channel. It'll be up within a week, and sort of fast forward until you get to that paper with the title of the paper.
Yeah, and if you get stuck, you can email me as well. either way.
Okay,
Yeah.
Nancy Honert has asked, are only basidiomycetes mycorrhizal?
No, there are mycorrhizal ascomycetes as well. Not as many. True truffles are a good example. Truffles are ascomycetes, and of course, truffles are mycorrhizal with trees. So yes, there are some cup fungi that are mycorrhizal. Some people claim morels are mycorrhizal. The word's out on that. There's not good, strong evidence that they are. Cup fungi, truffles, I think those are some of the main ones, but yes, there are some ascos that are mycorrhizal as well.
Cathy, there's a whole bunch of comments on here saying how terrific this presentation has been.
Wow, that's fun.
Our president, Patrick Plantenberg, says to all of us, thanks to all for attending. Please let MNPS know if you have ideas for future programs. So here's a question from Eric Godin. Is there any research or graduate projects pending for Glacier Park? As far as he knows, there's no one studying the fungi in the park.
The only thing I know there is that they've had several bio-blitzes, and the Western Montana Mycological Association, led by Larry Evans, has been involved. I was involved with one of the bio-blitzes, but they've had quite a few bio-blitzes in Glacier, and that Western Montana Club has come up with quite a list. So they are working. They're primarily amateurs, but they're at least working to get a species list. So yeah, they're at least cataloging the fungi.
Jennifer Sherry asks whether you know of any researchers who are looking specifically at some of the relationships that you mentioned between wildlife and fungi.
Oh gosh, I could reach around and pull a book off my shelf. There's quite a few, but there's a book called Truffles, Trees, and Beasts. And Truffles, Trees, and Beasts is partly written by Jim Trappy, who's done a lot of research on animals and fungi. So that's a little more academic, but Truffles, Trees, and Beasts is a good book if you're interested. So there are people working on that. They were working on it more in days past. I'm not sure. I haven't totally kept up with it. I sure see a lot of familiar faces. Roger Rosentretter, Robert Sheehy. My goodness. That's pretty neat. What are you doing in Montana?
It's the wonder of the World Wide Web, Cathy.
I guess it is.
Here's a question. How do allelopathic exudates affect mycorrhizal communities?
I don't know how they affect the communities, but allelopathic chemicals can move through mycorrhizal fungi. So while we're always thinking it's a benefit to have things moving through that web, allelopathic chemicals can also move through there. How they actually affect the fungus, that I don't know, that part of it.
What role do mycorrhizal fungi play in protecting their host from pathogens in the soil?
There's been a little bit of research on that, and they can, especially the ectomycorrhizal fungi, because they form that barrier on the outside of the roots. So they can provide some protection from pathogens. People have done greenhouse studies showing that if you have the right fungus and particular pathogens, that the mycorrhizal fungi prevent some of those pathogens. They can protect against little grazers in the soil because they have that layer on the outer part of the root. Mycorrhizal fungi also, I didn't mention all the things that they do. They also bind the soil. If you have a mycorrhizal system, if you pull up a tree, all of that root, this is hanging off the roots is being held there by mycorrhizal fungi. So they're holding the soil, and that makes it moister. I mean, a lot of different benefits of mycorrhizal fungi. I didn't even get into that part.
Ah. All right. More compliments, Cathy.
Boy, what a good group you have. What good question.
Someone has asked, Augustus Sauer has asked, are there ways that mycorrhizal fungi can combat climate change?
Well, if trees are beneficial against climate change using carbon, and mycorrhizal fungi benefit trees, I guess, by direct association. Mycorrhizal fungi are helping trees proliferate in landscapes where they wouldn't normally be. That one fungus, Coenococcum, really comes to the fore in drought conditions. So if we end up with drought conditions, we have more Coenococcum. It might allow trees and seedlings to maintain themselves on the landscape longer. So yeah, I think mycorrhizal fungi could certainly benefit, but it's through the trees that you're thinking about that.
Cathy, I think we're just about out of comments and questions about fungi. There's a whole lot of thank yous and compliments, and I'll just sort of amplify what these commenters have said. It's been just a really interesting and engaging presentation. Thank you so much.
You bet. And hi to everybody I didn't get to say hi to. I mean, I said Roger, Robert, oh, Peter Acuff's there. I don't have all the names, but thanks for everybody for coming too. Thanks for inviting me, Gretchen.